cytoskeleton and cell wall. Hashtag protect and provide. Welcome to our group project. Our team members introducing Sophie Pham, Jocelyn Powers, Emmanuel, I can't pronounce your last name, and Angel Rios.
Structures of a cell wall. The cell wall is a rigid, protective, and supportive layer that surrounds the plasma membrane in plants, fungi, bacteria, algae, and some archaea. Its exact composition depends on the organism. For plants, the primary cell wall is thin, flexible, made of cellulose, hemicellulose, and pectin present in young growing cells. Secondary cell wall is thicker and stronger, which contains more cellulose, lignin, and sometimes cutin, formed after cell growth stops. In fungi, is primarily of chitin, a tough polysaccharide. In bacteria, it's composed of peptidoglycan, a mesh of sugars and amino acids. It may contain a pseudopeptidoglycan, proteins, or polysaccharides. The functions of the cell wall serves many critical purposes: structure, support, and shape, protection, regulation of water balance, cell-to-cell communication, and growth control. What does the cell wall use? The cell wall uses polysaccharides and proteins to build and maintain the wall. Energy, ATP directly, since enzymes and transporters in the plasma membrane assemble wall materials. Water and ions to hydrate and maintain the gel-like matrix between fibers. What it provides for the cell: strength and rigidity, turgor pressure support, selective permeability, and defense. Organelles that depend on the cell wall. The plasma membrane, supported and protected by the cell wall. The vacuole in plants works with the cell wall to maintain turgor pressure. The plasmodesmata cross the wall to connect the cytoplasm of adjacent cells, linking their ER and plasma membranes. Chloroplasts indirectly benefit from the wall, since the rigidity and turgor pressure keeps the leaves spread out for maximum light absorption.
Now, moving on to the cytoskeleton. The structure: a dynamic network of protein filaments. Shape and support: gives the cell its structure, prevents it from collapsing, and anchors organelles in place. Movement: essential for cell motility, powers cilia, flagella, and ambioid movement, plus forms the spindle during cell division. Transport: works as the cell's highway system. Motor proteins like casein, dynein, myosin use ATP to move vesicles, proteins, and organelles along it. Organelles that depend on the cytoskeleton: the endoplasmic reticulum and Golgi apparatus, positioned and maintained by microtubules. Mitochondria and chloroplasts, moved and distributed with the help of microtubules and motor proteins. What it uses: ATP and GTP. Actin filaments require ATP for polymerization and movement. Microtubules require GTP to assemble and disassemble. Motor proteins, casein and dynein, use ATP to transport organelles, while myosin works with actin for contraction and movement. Why you should vote for us. Reason number 1: no shape, no strength, no survival. Without us, cells burst or collapse into blobs. No plants, no structure, no life. Reason number 2: no movement, no transport, no communication. We are the highways and connections. Without us, nothing moves or communicates. Reason number 3: no protection, no growth, no future. We shield cells, help them divide, and keep them alive. Without us, cells are defenseless. Why you shouldn't vote for the rest. They all depend on us. Without the cytoskeleton or cell wall, organelles can't stay in place, move, or even work properly. Number 2, they can't protect the cell. No cell wall means bursting. No cytoskeleton means collapse. Number 3, they aren't enough on their own. DNA, proteins, or energy are useless. The cell can't hold its shape or transport anything.
Thank you for watching. We have our group project table number 6, Sophie Pham, Jocelyn Powers, Emmanuel P, and Angel Rios. Thank you for watching.